Hi there. This is part 2 of the cancer presentation, mainly from chapter 18 in the Integrated Molecular Evolution second edition. In this part, I'll be talking about the molecular basis of cancer. So I'll be talking about mutations, recombination, those sorts of things, the things that lead to gene changes in the cells that lead to carcinogenesis. As I discussed in the first part, cancers are the result of several mechanisms that are also related to evolutionary processes. So there are point mutations. Some cancer genes can be inherited, recombination events, some of which cause chromosomal translocations, are very common in carcinogenesis. Gene amplification events are known in several types of cancers. Environmental exposures to mutagens, and then mobile genetic elements, including viruses, transposons, plasmids, and introns. To go to a few specific examples, BRCA1 and BRCA2 are two genes first identified in breast tumors, but they've been found in other types of cancers as well. On chromosome 17 is BRCA1, and I've already discussed that in the previous presentation, that's central to recognizing DNA damage and initiating DNA repair or turning on p53 to cause cell death. BRCA2 is a little more peripherally involved in that, but also involved in the same process. The BRCA1 gene is located on chromosome 17 in humans, and the BRCA2 gene is located on chromosome 13 in humans. Several studies mapped the point mutations in breast tumors for the BRCA1 and BRCA2 genes. The aim of the studies were to determine whether there were any characteristic mutations in those breast tumors. This is a graph of the frequency of those mutations. And you can see that the mutations are pretty much distributed throughout both genes. And so the conclusion was there was no specific hotspots for mutations in those genes. Furthermore, the red lines on some of these indicate mutations within the coding sequence, and the blue lines indicate those mutations that were in the intron-exon borders. Again, there were no real hotspots found either in the coding region or in the intron-exon borders that would be hotspots for cancer-causing mutations. In the BRCA genes, as well as other genes that have been studied since then, it's been found that if a gene is responsible or found as causing a tumor, the mutations can be found throughout those genes, and there aren't really any or many hotspots in those to cause cancers. In addition to point mutations causing certain types of cancers, human chromosomal translocations are also indicated in cancers as well as some other diseases. So at the top, a translocation between parts of chromosome 2 and parts of chromosome 5, they exchange pieces, and that's known to cause anaplastic large cell lymphomas. Burkitt's lymphoma is a translocation between chromosome 8 and chromosome 14, and then there are other chromosomal translocations noted here, including one for Down syndrome, which is a translocation between chromosome 14 and chromosome 21. And so that type of Down syndrome doesn't involve trisomy of chromosome 21. It involves a chromosomal translocation that interrupts the genes. This is another representation of those chromosomal translocations, indicating the translocations between specific chromosomes. So at the top, there's chromosome number 22, which has a translocation with chromosome 3 to cause non-Hodgkin's lymphoma, chromosome 22 with a translocation to 17 causes a different type of tumor, and this is shaded so that the chromosomes that have the most chromosomal translocations involved in cancer are in black, and then ones that are somewhat involved in translocations that cause cancer are in lighter gray, and then the ones that are still white are chromosomes where translocations haven't been identified that cause any tumorigenesis or carcinogenesis in humans at least. Here are a couple of examples of some of the things that can happen and do happen. So if you have copies of gene A, they can cross over where they're not exactly lined up with one another, having a crossover within an intron region. And then you can get two new versions of the gene A prime and gene A double prime, which may have no function at all, maybe non-functional, or they can have an altered function that can cause aberrant proteins and reactions to occur within the cell. The second thing that's common is you have two non-homologous genes and you get a crossover between them. And usually when this involves carcinogenesis or tumorigenesis, the promoters and enhancers can be exchanged, and so you can have promoters that have very limited expression patterns. Suddenly that gene now is turned on constitutively or turned on all the time, and conversely you might have a gene that's turned on constitutively or very often, and now it's not turned on to the same extent, and those can actually have quite large effects depending on what type of protein they form, whether it's in signal transduction or whether they're receptors, etc. Here's one example that where a translocation causes Burkitt's lymphoma. These are tumor cells of B lymphocytes. So you have a crossover between chromosome 8 and chromosome 14. They exchange pieces, creating a translocation of chromosome 8 and chromosome 14. And in the breakpoints, that interrupts genes or changes the gene expression of some crucial genes that control cell replication and division. And Burkitt's lymphoma is the second one there, where you exchange a promoter on the MYC gene, and the MYC gene is a transcription regulator. So instead of the MYC gene being turned on only during certain times in certain cells, now with this promoter, which came from chromosome 14, and the MYC gene's on chromosome 8, as you can see there, now the MYC gene is turned on at much higher levels and is turned on all the time, which creates the tumor cell. Also there is the follicular lymphoma, where there's a translocation between chromosome 18 and chromosome 14, where now the BCL2, which is another breast cancer-like protein involved in signal transduction, is put next to a very active enhancer, turning that gene on. The third one down causes a diffuse large B-cell lymphoma, which is a translocation between chromosome 14 and part of the immunoglobulin gene family, and BCL6, another breast cancer-like protein from chromosome 3, causing that lymphoma. And then a prostate cancer where the ERG, which is an estrogen receptor gene, is put next to a very active promoter causing a prostate cancer. And finally, the chronic myelogenous leukemia is caused by a translocation between chromosome 22, which has the BCR gene on it, and chromosome 9, which has the Abelson gene on it. And remember, the Abel was a signal transduction protein. So now, instead of being turned on to a limited extent, it's turned on constitutively, and that causes the tumor cells and cancer. This diagram has some similar changes, just at the top is just indicating it's sort of a general situation where you're switching around promoters and enhancers onto genes that are normally highly controlled. Now they're constitutively expressed, so they're turned on at much higher levels. So there's one, they're the follicular lymphoma caused by a crossover and translocation between a BCL gene and part of the immunoglobulin gene. B is a Burkitt's lymphoma, again turning on the MYC gene constitutively. C is a diffuse large cell B cell lymphoma, where the BCL gene is turned on to a much higher extent than normal. And then the prostate carcinoma turned on by a highly active promoter region. These mutations, these translocations, can be initiators of the tumors or can be several mutations down the road from the initial mutation. But remember, as I was talking about in the presentation last time, cancers progress through many mutations until they become more dangerous all the time, being vascularized and eventually invading tissues and metastasizing. And that takes quite a few different mutations together. So this shows some very specific translocations in Burkitt's lymphoma. It turns out that there are many different crossover points in those translocations, and the arrows indicate those. So it's between a gene on chromosome 8 and another gene on chromosome 14. And so you can get crossovers, combinations of crossovers in each one of those places where the arrows are to create a new gene that, instead of the MYC being expressed to a very limited extent in only certain types of cells and tissues, now the switch turns that gene on to much higher extents, and then that causes increased transcription and movement towards a tumor and a cancer. This also shows that even the Burkitt's lymphomas can have slightly different mutations and translocations, so each cancer is somewhat unique. Here is another example, this one for chronic myelogenous leukemia, and this has chromosomal translocations between chromosome 9 and chromosome 22, and this affects the BCR and Abelson genes, ABL. So you get crossovers in any of those regions in the arrow. So you see those are all in intron regions, which is fairly common. So instead of the Abelson gene being turned on to a small extent and very controlled in certain cells, now it's turned on to a high extent, and that changes signal transduction pathways within the cell and turns them into tumor cells, forming cancers. Here's another example, this for small cell lung cancers. Now these cancers are some very dangerous lung cancers with high mortality rates, but you can see that the main part of this, the ALK, which is a tyrosine kinase section of the eventual protein, is joined together with active promoters, but they can be different versions of those active promoters. So in this case, the proteins are produced in high levels where normally they're not produced in very high levels, and they can phosphorylate other proteins and activate those proteins. So in this case, they can activate these proteins to cause the cell to become a cancer cell where it grows and divides rapidly. Here are some other examples, all of which have this bromodomain in the proteins. And the bromodomain acts on other proteins to activate them. So you can see that you can have this bromodomain switching into various different chromosomal locations to cause activation of those genes and proteins that are ultimately formed in those cells. This represents a summary of looking in the vicinity of the KRAS gene in cancer cells. Remember, KRAS is that one that, when it's activated, will activate that MAP kinase pathway and stimulate transcription of quite a number of genes. So what they found was, in addition to the interchromosomal changes, that is, chromosomal translocations, they also found head-to-head recombination events, tail-to-tail recombination events, tandem duplications of sequences, and deletions of sequences in that region, all of which had an effect on the expression of KRAS, which would then affect the control of transcription in those cells. Each of these on the previous slide can be created by a recombination event. So by looping out, you can get a deletion on the chromosome, sometimes small deletions, sometimes quite large deletions. You can also get duplications. Some of this can be caused by during replication, but also transposable elements can duplicate regions on the chromosome. And also, if two chromosomes line up differently, one can have an insertion and one can have a deletion. You can also get inverted regions. These are normally created during replication also by breakage and rejoining of the chromosome as it's being replicated. Insertion events can occur too, so you can have non-homologous chromosomes crossing over not just once to create a translocation as on the bottom, but you can have two crossover events and then get a chromosome piece inserted into one chromosome and deleted from the other chromosome. So all of these are things that you saw from the previous slide when you got all of those rearrangements around the KRAS gene. So here are a few other possible rearrangements similar to the previous slide, in that you can have homologous recombination, that is, two homologs can line up side by side and you can get a crossover between those. Now, if they have different alleles on them, then the new chromosomes still homologs, but they can have different allele arrangements. You can also have chromosomal inversions where you have a crossover in different locations on the homologous chromosomes, and invert sections of both chromosomes. So then you've got an inversion on both of those chromosomes. You can also have homologs misaligned during recombination. We've already talked about that with respect to ribosomal and other highly repetitive regions. This can happen, but it can also happen on a longer range, so that you have them misaligning. You get a crossover, one chromosome has a duplication, major duplication, and the other one has a major deletion in the chromosome. And then finally, between non-homologs, you can get crossovers to create chromosomal translocations. All of these chromosomal rearrangements are capable of creating mutations that will cause a cancer. Another set of events that can cause cancers or to make cancers worse are gene amplification events. So in the case of the somatostatin and angiotensin-like peptide receptors, in lung cancers, certain lung cancers, the amount of amplification of the DNA is 5.5-fold, but the overexpression of the protein can be as much as 247-fold. In colon tumors, you can get an amplification of an average of 7.6-fold, and over 1,000 times fold of overexpression. Ovarian cancers, you can have a 7.5-fold increase in the number of DNA copies, and about a 7.2-fold increase in the overexpression of those genes. And then pancreatic tumors, 2.6-fold in the DNA and 137-fold increase in the overexpression of the protein. For CTSZ genes, which encode proteases, a certain protease, in colon cancers, maximum fold is about 12 times, 12x meaning 12 copies of those genes, and up to a 52-fold increase in the amount of protein that's produced. In breast cancers, a 5-fold increase leads to a 10-fold increase in the overexpression of the protein, and a 3-fold increase in ovarian cancers can create up to a 10-fold increase in the amount of those proteins. And all of these are known to be a part of tumorigenesis in those particular cells and cell types. This is the same data, same information, just put in a graphical form. So you can see for the somatostatin angiotensin-like peptide receptors, you can have between about a 2.5-fold to a 7.5-fold increase in the gene copies in those cells, but up to a 1,000-fold increase in the amount of protein that's being expressed from those genes. The cathepsin Z protein, which again is a protease, you have from a 3-fold to a 12-fold increase in the number of genes, the amplification of those genes, and up to a 50-fold increase in the amount of that protease that's being formed from those genes. Those increases, that amplification and those increases can greatly affect how aggressive the tumor is and how successful treatment is for those patients. So this is a graph of HER2 genes in breast cancer. And it shows that if there's a single HER2 gene, the solid line, then survival stretches out to about 50%, about 24 months of survival, and some live, 5 years or more, as long as they have only a single copy of the HER2 gene. On the other hand, if that mutated HER2 gene is amplified, then the survival at the 50% survival level is after about 10 months. So, and after about 15 to 20 months, then survival is almost zero. So you can see that if the gene is amplified, the tumor is more aggressive and the survival possibilities are lower in those patients. As I mentioned in a previous presentation, viruses can change DNA. They can mutate the DNA to the point, in this case, to causing a tumor, transforming the tissue. So in many viruses, you can have a latent infection. This happens with some herpes viruses where they form a little space for themselves in the cell, and then occasionally they will pop back out and cause infection and symptoms. On the other hand, you can get persistent infection where the virus stays in the cells and continuously sheds new viruses to infect other cells. HIV is known to do that. In some cases, the viruses will lyse the cells and kill them. And then in a different case, at the top, you have viruses that can integrate into the chromosome and transform those cells into cancer cells or virus-producing cells as well. Here's an example of a virus that carries an oncogene with it, that is, a mutated copy of a normal cellular gene, in this case Src, which is part of the signal transduction system that I talked about in an earlier session. So this virus can infect a cell as a reverse transcriptase. It's an RNA virus, of course, makes a DNA copy, and the DNA copy can be inserted into the host cell, or it can just simply go through transcription and translation, make its proteins, and bleb off more of the virus particles. If it does integrate into the chromosome, it's carrying the mutated SARC gene, the oncogene, and it will then start being transcribed, acting in the signal transduction system, turned on actively, and that will stimulate transcription of other genes, creating a tumor cell or a cancer cell from there. So there are actually two ways that this can happen. So on the top, you have a proto-oncogene, that is a gene that has the potential to become an oncogene in the cell and transform the cell into a cancer cell. So in one case, the virus can pick up a proto-oncogene, and it's actually the oncogene type, because it actually changes, and I'll show you that in a few minutes. And then that integrates into the chromosome of the host cell and transforms it, because now it has the oncogene, which will affect production of that particular protein product, whether it's a transcription factor, part of the signal transduction pathway, etc. Along the bottom line, what can also happen is the virus may carry with it a promoter region or promoter gene, which then integrates upstream from the proto-oncogene or the cellular oncogene and turns it on uncontrollably. So again, that would increase expression of certain genes like MYC, FOS, or JUN or something like that, or the RAS gene, and cause the cell to become a cancer cell. So here are a couple of examples of cellular and viral oncogenes. So on the top, in purple, that's a viral JUN, that is a transcription factor, and the c-JUN down below is the normal cellular proto-oncogene. So you can see the structure of those genes. The colors represent different domains of those particular proteins. The different sections of the normal cellular oncogene or cellular proto-oncogene, and then up above, the regions of a viral version of that are missing. So there's parts of introns missing and parts of a few exons missing, and that causes that particular transcription factor to be more active than normal. Then the lower ones are the FOS genes, the viral FOS and the cellular FOS. And again, the cellular FOS is down below, and then you see the viral version where the last 3' end, or the carboxyl end of the protein, is completely missing, has been replaced by another fragment, which again causes that transcription factor to malfunction. Here are some other versions of viruses that are carrying oncogenes. So up on the top, that's a normal ALV virus, an avian virus, and then the MULV is a murine or mouse virus with the long terminal repeats that you've seen before in both retrotransposons and retroviruses. Then you have an AMV, which is carrying an oncogene, and it carries a MIB gene, which is also a transcription factor. But note that the MIB, the cellular version of MIB, has 15 exons and 14 introns, and the MIB that's in the virus has no introns whatsoever. And this is pretty much the case for all of the other oncogenic viruses. The Rous sarcoma virus is there. Again, the C-SARC versus the V-SARC gene. The V-SARC gene has none of the introns that the cellular version has. And then the murine leukemia virus, the same. It's missing the introns, and you go to all of the others down the line. And in each case, the cellular proto-oncogene has a number of introns, whereas the viral version has no introns, and sometimes, which isn't shown, even the express version, the version that has the coding sequence in it, is also changed in some of those. Some viruses that cause cancer and transform cells integrate into the host genome, but they don't carry a protein encoding gene. They carry a gene that makes a microRNA. So the microRNA causes some changes in expression of the genes of the host. In fact, they can stop certain RNAs from being expressed, and this can also transform the cells. One form of cancer that's very prevalent in human populations is one of endometrial cells, that is uterine cancers, and also cancers of male reproductive organs also. And it's caused by the human papillomavirus, HPV. And so up above you can see that as it enters the cells, it begins to change the nuclei of the cells, that is, transform those into cancer cells. And it's very efficient at doing this, so that it can cause cancers quite readily in both male and female reproductive organs. However, there's a vaccine against the human papillomavirus, and if you get the vaccine early enough so that you are never infected with this HPV, then these cancers are almost non-existent in males and females after the vaccination. One set of molecules that can greatly affect what happens in tumorigenesis and the progression of cancer are hormones. So hormones can stimulate cells, but they can also stimulate the cancer cells if they happen to be there. Occasionally they can stimulate the initiation of cancer, and they can also stimulate the progression of the cancer. They're all pretty much related. So you start up here with cholesterol. Just one enzyme can change it into pregnenolone, and then a set of other enzymes can produce progesterone, cortisol, estradiol, testosterone, dihydrotestosterone, aldosterone, corticosteroid, etc. So you can produce a number of both male and female hormones, starting from the cholesterol molecule. Estrogen turns out to be pretty potent in its activity with regard to stimulating cancer cells as well as other cells. So it has one effect that's direct. Estrogen interacts with an estrogen receptor, and it goes actually straight to the nucleus. It's not acting at the membrane level. And it can be a transcription activator in that case. It can also be tethered to other transcription factors and increase transcription of certain genes. It can act in the cytosol as part of the transduction pathway. And it can be ligand independent. So the estrogen receptor, if it's phosphorylated, when it's activated by protein kinases, it can also act as a transcription factor and increase transcription. So it's pretty varied and very, very active at promoting transcription in cells. It can affect uterine cancers in a couple of ways. One is that it can stimulate division of existing mutant uterine cancer cells. And so it can increase the size of the tumor by increasing cell division and activity of those cells. It's also been noted that because it increases the number of replication cycles and cell divisions, that it can increase the rate of spontaneous mutations and then cause a cancer in that way. Although estrogens can increase the rate at which tumor cells grow, so they're in the adipose tissue produces estrogens, the ovaries produce estrogen, and there are actually plants produce phytoestrogens, things that act like estrogens. And then there are some drugs like DES and other things for birth control, estrogens. But also other types of chemicals like BPA in plastics, prolactins, and progesterones, and growth hormones from pituitary can affect the growth of tumors once they're initiated. So when women become pregnant, sometimes they find out that a tumor that they didn't know they had suddenly starts growing much more rapidly, and that's because of the boost in hormones during that time. So not only estrogen and estrogen receptors, but then there are receptors on the outside of the cells, such as prolactin receptors, that will increase phosphorylation of proteins, increase the MAP kinase production through RAS, and also increase other transcription regulation factors. So those can affect motility, survival of the cells, proliferation of the cells, and differentiation. All of these then, if that receptor goes bad or the cell happens to have mutations that cause a cancer, then the prolactin and other hormones actually can stimulate those cells to grow even faster than otherwise. So just to reiterate, then I talked a little bit about point mutations and how they're sort of unpredictable when they happen. Inheritance is a little more predictable as long as you have the genetic sequences or at least a characterization of the family. Recombination events, especially those that affect chromosomal translocations, gene duplications, amplifications, things like that, and then gene amplification, of course. Environmental exposures to things like plastics and plant hormones and things like that. And then finally, genetic mobile elements, including viruses, but also there are other mobile genetic elements that can rearrange different parts of the genome and cause cancers. Finally, this just shows the changes in cancer rates from 1988 to 2008, so probably should update this, but it hasn't changed all that much. So certain cancers are decreasing in their incidence, stomach, colorectal, ovary, lung, and most of these are due to increased surveillance, stopping of smoking, and ceasing from other activities. But then some of the incidences are going up, such as pancreas, prostate, lymphoma, melanoma, and thyroid. Some of them, like melanoma, is related to sun exposure. Pancreatic cancer is going up, and it's quite deadly, so no one knows exactly why that one's going up. So in general, the rates of cancers, if you sort of even them out over all cancers, the incidences have been sort of remaining steady, although the death rates have been going down for most cancers, and that's due to better treatment, especially now with immunological therapies that have proven pretty successful for many different types of cancers now. Okay, thanks. This is the end of the 2 cancer presentations, and next I'll be talking about basic genetics, talking about what a species is, and then doing also some phylogenetic analysis presentations as well. Okay, thanks.